Okay, so this morning we are still continuing our conversation on microbial diversity. We've wrapped up our talk about viruses and other acellular agents, but today we're going to take a look at the domain bacteria. All right, so what do we know about bacteria? There are many pieces of information that we've continued to discuss throughout this semester. Today we want to take a quick look at some details in terms of bacteria as far as their morphology. Morphology refers to the shape of the bacteria. You've probably, if you're in lab with me, we did a exercise on last week or the week before where we looked at bacteria morphology under the microscope via staining. So we're going to talk about that process a little bit more today, how we can examine bacterial morphology. We'll look at some reproduction in bacteria. This is going to be a little bit different than what you see in other organisms. It's a form of asexual reproduction. We're going to look at, again, some bacterial staining procedures. And this is a very important tool or technique as we look at microbial diversity, because this staining allows us to sort of better visualize bacteria. All right. So again, if you're in the lab, we've talked about how bacteria appear sort of clear or translucent in nature. So we have developed various staining techniques to be able to better visualize various properties of our bacteria under the microscope. And then we'll look at some other characteristics of bacteria and some unique bacteria that do not conform to all of the general properties that we'll talk about in general. Okay, so let us get started. So we know that this domain bacteria has the feature of being prokaryotes, right? We know features that define the prokaryotes are that they do not contain a nucleus and other membrane-bound organelles, right? We know that all of the organisms in this domain are going to be unicellular organisms, right? So first and foremost, these are very basic identifying characteristics of bacteria. They're prokaryotic, they don't have a nucleus, they don't have other membrane-bound organelles. We know that they do have structures common to all cells. These are living things, so they are cells. They do have a plasma membrane or that biological membrane that we've talked a lot about already. And then bacteria also have an additional barrier of protection around it known as the cell wall. All right. But again, we're saying that these organisms are unicellular, which means that they contain only a single cell. And that one single cell has all of the ability to carry out all of the properties of life, right? So we, again, in your biology class, you established this set of criteria for determining if an organism is living or not, right? What were some of those criteria? Can you recall any of them? There were about 7 or 8 properties that an organism must exhibit in order to be considered a living thing. This unicellular organism exhibits all of those. Can you recall any of those properties? What are some characteristics of life, guys? Anyone? I'll wait. Can anyone identify any of the characteristics of life?
The ability to metabolize, isn't that one of them?
Yes, metabolism is absolutely one of them. Very critical one. This is how we get energy and use energy, right? Metabolism refers to the total sum of all of the chemical reactions that's going on in a living thing, right? We are constantly undergoing metabolic activities. Our bodies are just a big ball of chemical reactions. What else? Yes, metabolism. Let's see.
Reproduction.
Chat. Yes, reproduction. Absolutely. We know that reproduction is a requirement for life. Bacteria undergo a special form of reproduction known as binary fission. All living things must be able to reproduce. Tara said growth and development. Yes, absolutely. All living things must have the ability to grow and develop, and that is absolutely a requirement for life. Tashiana says homeostasis. Absolutely. So the bacteria cell, though it is a very simple cell type, it doesn't have all those fancy membrane-bound organelles like mitochondria and so forth. It can carry out all of the properties of life individually. So maintaining homeostasis, maintaining that constant internal balance, right? Responding to stimuli, absolutely. Responding to stimuli. So anyway, we're here defining this domain, and you should be able to identify for me some of these characteristics of life. All right. So in terms of bacteria morphology, all right, morphology again is referring to the shape of the cells, right? We can categorize or identify bacteria based on the shape of their cells. If you don't have these if you haven't jotted these down in your notes, jot these terms down: cocci, bacilli, and spirillum. Okay, these are three general morphological shapes of bacteria. All right. Cocci are typically going to have this spherical or circular makeup. These cells are going to appear sort of like a sphere or a circle. Okay. Cocci. All right. Bacilli, these are going to be our rod-shaped bacteria. Okay. Bacilli are rod-shaped. Okay. So it looks like this thin rod as opposed to this circular sphere shape. Okay. And then we have the spirillum. Okay. Spirillum sort of present with this spiral type shape. Okay. Curved, spiraled arrangement. All right. You should be familiar with bacterial morphology. You cannot leave this class and not be able to have some basic understanding of bacterial morphology and various arrangements of bacteria. Okay. So cocci, bacilli, spirillum. Okay. These are categories of bacteria based on the shape of their cells, right? And so as we get to our conversation about staining, this is what these staining techniques allow us to better visualize these characteristics of bacteria, right? And so once we can determine if it's a sphere or a cocci, bacilli, spirillum, we can then further examine how these cells are arranged together, how are they organized together, right? So bacteria can have various arrangements. This slide right here shows you some arrangements of cocci, all right? And some of the examples of organisms that have this arrangement, okay? So diplococci means that we've got two pairs of cocci. They're gonna appear in pairs, okay? Diplococci. So an example of a bacteria that has this morphological arrangement is Neisseria gonorrhea. What do we know about this organism? What do we know about Neisseria gonorrhea? Does this name ring familiar for anyone? All right. So Neisseria gonorrhea is the etiological agent that causes gonorrhea. This is a sexually transmitted disease that can actually be very problematic if left untreated. All right. So diplococci, this is the organism that can appear in the morphology is cocci, but they appear in pairs. Okay. Streptococci. All right, strepto, we're talking about some chains of cocci. So we see chains, okay, linked together in short chains. All right. An example would be Streptococcus pyogenes. These actually cause strep throat. right? You or someone that you know have probably had strep throat before, okay? Streptococci, okay, that's the arrangement. The morphology is cocci. The arrangement is sort of in these chains, all right? So Streptococcus pyogenes. Staphylococci, all right, these are clusters, all right? These are cocci that appear in a clustered arrangement, all right? Staphylococci, Staph aureus. This is something that's very common. It causes these boils that you might have had on your skin before. You may know someone that's had a boil. They're very painful. They're caused by Staph aureus. Okay. Tetrads. Okay, we got these 4. Okay, tetra. Okay, we have 4 arrangements of cocci in groups of 4. All right. Micrococcus luteus. This is rarely pathogenic. This is a very common bacteria. You actually find Micrococcus luteus. You can find it in your bathroom. Like if you've ever seen, it's not a good thing if you see it. Means that you're not cleaning your bathroom enough. But if you let your sink or bathtub go too long without being cleaned, you'll start to see kind of a pink film. That's Micrococcus luteus building up around your bath area. It's not really pathogenic so long as you have a intact immune system. As long as you're not immune compromised, Micrococcus luteus is not going to hurt you. Octads, okay, we've got groups of 8, okay. Sarkinia, these are definitely all examples of arrangements of cocci, okay. We've got diplo, strepto, staphylo, so we've got clusters, we've got chains, we've got pairs, we've got tetrads, right? So again, these are morphological arrangements that we can see as we stain our bacteria, all right? Reproduction of bacteria, you've probably discussed this a little bit before, but the process is known as binary fission. All right, jot this down. Binary fission is a form of asexual reproduction in which an organism divides into 2, each part carrying 1 copy of genetic material. All right, so how does this differ from sexual reproduction? How does binary fission, how does binary fission differ from sexual reproduction? How does asexual reproduction differ from sexual reproduction? That is a question that I'm waiting for an answer for.
With sexual reproduction, you need a mate.
Right. Sexual reproduction requires 2 parents. Okay. If you don't remember that, jot that down. You should be able to have that point of comparison in your mind, right? With binary fission, it only requires 1 organism. Okay. This organism will undergo mechanisms to duplicate its genetic material, the DNA. Okay. So we start with the parent cell. We're going to duplicate the genetic material. All right. And this cytoplasm will ultimately divide and yield 2 daughter cells that are going to be exact replicas of the parent cell. Okay. Each new cell that's created will have 1 copy of the genetic material. And we know that in bacteria, that genetic material is a single chromosome, right? Single chromosome. What questions do you have about that? We're all familiar with bacteria reproduction, okay, and the process of binary fission. If not, I do admonish you to go out and read a little bit more so that you understand what's happening and how this process is different than what happens with sexual reproduction, which requires 2 parents and those gametes, the formation of those gametes, the sperm and egg cell, unite to form a zygote. Right, that's what we see in our higher ordered eukaryotic organisms. But in bacteria, we're seeing binary fission as a form of asexual reproduction. Okay, in fungi and yeast, we see asexual reproduction in budding and fragmentation. Right, so those are other examples of asexual reproduction. Staining. All right, so as I mentioned before, bacterial staining is absolutely crucial in us being able to explore and understand the diversity of organisms, right? I mentioned before that most bacteria exist in nature in sort of a colorless or translucent color, and they're difficult to see. All right, they're clear, they're sort of translucent. They're very difficult to see under the microscope without stains. All right, so staining techniques allow us to better visualize bacteria. All right, and so before we can actually do these stains, we have to create what we call a smear prep. Okay, write this down. I do want you to be familiar with what's necessary for us to stain. Okay, a smear prep of bacteria is created on a microscope slide in preparation for staining. All right, so we would obtain our bacteria from whatever growth medium we're growing it on, be it agar or broth, right? If we're in the lab, we've already talked about media that we use to grow bacteria on. And if you haven't taken the microbiology lab yet, you will, right? Microbiological media are basically the nutrients that we grow our bacteria in, right? So you've seen Petri dishes before. Those Petri dishes contain the semen solid that we call agar. And those agar are basically a composition of nutrients and substances that these bacteria require to grow. So anyway, we would grow our bacteria in some nutrients, right? And the broth, it's a liquid medium, right? That we would see, like if you saw bacteria grown in a flask or a test tube, that might be your liquid broth. And then if it's on a plate, right, that Petri dish, that's our agar. So anyway, we grow up our bacteria, and then we're actually going to have to extract some of it from whatever growth medium that we had it on in order to create this smear prep, right? And so once we create this smear prep, again, which is basically us extracting some bacteria from whatever growth medium we're growing it in, and then we place it on a slide, on a microscope slide, and then we have to do a process that we call heat fixation. Okay, jot this down. Once the smear prep is created, the smear prep is merely us taking some of the bacteria off of that Petri dish and smearing it onto a microscope slide, right? That's our smear prep. And then we do a process known as heat fixation, all right? Heat fixation is essential before we can actually do our staining procedure, okay? Heat fixation serves three purposes, right? One, it kills the organism. We don't want our organism moving all over the place if it's motile, and we'll talk about some of those properties in just a moment. Some bacteria can be motile. We want to make sure that we keep that bacteria in one place on our slide. Heat fixation allows us to sort of preserve the morphology of the bacteria as well, right? So that's one of the things that we're probably interested in if we're staining. We want to observe this bacteria in its natural existence. So we want to see morphology. We want to see the arrangement, right? So basically, we are using this heat to anchor this bacteria in one place on this slide. All right. I want you to be familiar with that and be able to describe for me what's happening with heat fixation and why is it important. All right. Heat fixation is a very critical second step after we create our smear prep in order for staining to take place. All right. So we would basically have a microscope slide with bacteria on the top, and then we would use our Bunsen burner and we would wave that slide sort of through our flame 3 quick times. And we did this last week in the lab, if any of you guys are in lab with me. We did a smear prep and a stain last week. So anyway, bacteria staining procedures. Why do we do these? Different staining techniques can be used to observe various characteristics of our bacteria cells. So again, morphology, we can see our size better. We can see the arrangement a little bit better. We can see the cell wall composition a little bit better. We can see other structures that may be present on the cell, such as the capsid, the flagella, right? What's the purpose of the flagella on the bacteria? What's the flagella used for, guys?
Mobility.
Yes, motility, locomotion. This flagella aids in movement. All right. What's the purpose of the capsule? We talked about that a little bit earlier in this semester. What's the purpose of the bacteria capsule? Does anybody remember? All right, so it basically kind of provides another layer of protection from the for the bacteria. All right, it can confer some resistance to antimicrobial agents and so forth. So this capsule is not found on all bacteria, but some bacteria have this additional barrier protection outside of the cell wall. And then we know that bacteria have the cell wall, right? This is a barrier of protection just outside of the cell membrane. All right. So we use bacterial staining for purposes of being able to better examine our bacteria. All right, so one type of stain that we do is called a simple stain, right? Simple stains basically are used to determine morphological arrangement, right? It's just a quick way to add some color and look at sort of morphology, all right? So we're using a single dye, right? That's going to be added to this smear prep, right? So if you look at this picture here, I have a schematic of the process from start to finish. You use the inoculating loop. Can you please mute your mic? Thank you. So a simple staining procedure starts with us creating a smear prep, right? So we're going to smear a loop full of the microorganism onto our microscope slide. And this tool right here is an inoculating loop. Write that down, right? So we use an inoculating loop to extract some bacteria from our plate, and we smear it onto this slide, right? We're going to allow it to air dry. In this illustration, they're using a methanol fixation, but we typically just use heat fixation in our lab. And then you're basically going to flood your slide with the stain, with one of your simple stains. All right, this simple stain is going to be of a particular color. We, flood the slide with the stain. You're going to add, so you're going to use your dropper, right? You use your dropper to add several drops of your dye onto your prep, your smear prep, right? After about 20-30 seconds, depending on what dye you're using and what your instructions are, you're then going to rinse that dye off, right, with water, blot it dry, and then you're going to examine your slide under the microscope, and we are going to use oil immersion, right? And again, if you're in lab, we learned that technique last week, and if you haven't, you will when you take lab. Okay, so I want you to be able to sort of describe for me what's happening for a simple stain. All right, another type of stain that you might be familiar with is the gram stain technique. This is what we did last week in lab. All right, the gram stain technique is a method to quickly sort of differentiate or classify bacteria. And the gram stain technique is based on the structure or composition of the bacteria cell wall. Write that down. That is very important. The gram stain technique is based on the composition of the bacteria cell wall. Every single bacteria cell has a cell wall, right? But some bacteria have a thin cell wall and some bacteria have a thick cell wall. Now, what's the major component of this bacteria cell wall here? I know we've talked about it before. What's the major component in a bacteria cell wall?
To help it maintain its shape and structure?
Yes, no, that's the function of it. What's the major component, like the makeup, the composition? What chemical makeup is there? You guys remember the peptidoglycan? We talked about this a little bit. Jot this down. The bacterial cell wall is made up of this peptidoglycan. Write that down if you don't remember it. Okay, this is the major molecule that's found in the cell wall of a bacteria. All right, it's sort of a polysaccharide with peptide, like proteins and sugars. All right, the gram stain procedure, real quick. There are 4 reagents that are required for the Gram stain procedure. Okay, so again, remember, this is a technique that's going to allow us to differentiate or classify bacteria quickly based on the composition of that cell wall, whether there's a thin peptidoglycan layer or a thick peptidoglycan layer. So the 4 reagents that we need for the Gram stain are crystal violet. Okay, this is our primary stain. Iodine, which is going to function as a mordant, right? The mordant has the ability to sort of fix this stain into the cell, all right? It's trying to help this crystal violet be uptaken by the cell. And then there's alcohol. Alcohol serves as a decolorizer in this procedure, all right? And then safranin is our counter stain, all right? So we're using 2 stains here, all right? Crystal violet and safranin. And so again, once we have created our smear prep, as we talked about before, we heat fix the bacteria onto the slide. We're going to start our procedure by flooding the slide with this crystal violet solution. Okay, this crystal violet solution is going to appear sort of a very dark violet purple. All right, so you're going to flood your slide with crystal violet. You're going to rinse your slide a little bit with water, then you're going to add your iodine solution. Okay, your iodine solution again is functioning as a mordant. It's trying to assist these bacteria cells with taking up that crystal violet dye. All right, then we're going to rinse off our iodine and we're going to try and decolorize. In this illustration, they're using acetone, but we use alcohol in the lab. Decolorize, we add some alcohol, let it sit on the slide for a little bit, wash the slide, and then we apply our counter stain. All right, safranin. Safranin is the counter stain. Now at this point, depending on whether or not, well, actually at the point of decolorization, depending on whether or not this is a gram positive or gram negative cell, these cells will either appear purple or clear. They would have been decolorized. Okay. The alcohol being added will, in a gram negative organism, will rinse off these cells and they will appear clear. And when we add our safranin, which is a counter stain, these cells will take up this safranin and appear a sort of a red pinkish color under the microscope if it's gram negative. However, if this is a gram positive organism, it's going to have a very thick peptidoglycan layer. And at the point of decolorization, this decolorization process does not work. And these cells will retain the crystal violet color, this purple color. All right. And so adding this counter stain will not change the color in which these cells appear. And then we would visualize it under the microscope and we would either see cells that appear purple or violet, or we would see those that appear sort of a pinkish red color. All right. And that is how we would interpret or categorize our bacteria cells based on the result of the Gram stain procedure. All right, so it's a very simple procedure where all you're doing is adding a few drops of dye and rinsing, and it takes about 5 minutes to do. And again, it's a very useful tool in microbiology. It's probably one of the most used tools in microbiology for quick identification or classifying of an unknown bacteria. We create our smear prep again, where we're just going to smear some bacteria onto our microscope slides, and then we're going to flood it with some series of dyes and stains and kind of see what happens at the end. So real quick, the analysis, how do we interpret the Gram stain? All right, if your organism is Gram positive, again, we're saying it's going to have a thick peptidoglycan layer and it's going to stain sort of a purple bluish color under the microscope. All right, so we can look and start to see this cell a lot better. We can see this morphology right here. What morphology do we see right here? What will we call this shape? We just talked about this today. What shape is this here? What shape is this? Someone. What shape are these cells that we stained gram-positive here?
Circles?
Say it
Is it
again.
oxide?
Yes, those are cocci. These are gram-positive cocci because they appear sort of a circular spherical shape. Absolutely. All right, and then if it's gram-negative, then this organism has a thin peptidoglycan layer, and it's going to stain sort of a pinkish reddish color under the microscope. All right, guys, you need to definitely take some time and review this information. I do want to go ahead and make an announcement. We're going to go ahead and have our third exam on Monday. Monday, we're going to have our third exam on microbial diversity. So we're going to continue on Friday and wrap up this and kind of have a quick review, but we need to take our third exam on Monday. All right, make a quick note of that. I'm going to stop right here today. We still have a few more things to talk about here with bacteria, and then we'll talk a little bit on Friday about